Hello. Welcome to the step-by-step tutorial. In this tutorial, you will learn how to analyze different data types supported by PaintOmics. We developed PaintOmics, a web server for the integrative analysis and visualization of any combination of omics modalities, using biological pathway maps. Basically, PaintOmics takes a list of omics datasets together with lists of significant features and paints this information on KEGG, reactome, or MapMan pathway maps. Pathway enrichment analysis, pathway interaction network, metabolite hub analysis, metabolite class activity analysis, metagenes, and regulatory omics analysis methods are implemented in the application. The video contains 4 sections. Section 1 includes the introduction of the PaintOmics layout. Then, Section 2 includes a guide on using PaintOmics to analyze gene or metabolite-based omics data, which covers omic data types that can be translated into genes or assigned to metabolites. The following section is region-based omics, including all those omic data types where the information is grouped around a set of genomic locations of interest. The final section is regulatory-based omics containing any trans-acting element operating on genes, transcripts, or proteins. Now, we start by looking at Section 1, Interface. First, let's look at the left sidebar. Job View allows you to return to the current job page. Personal storage has several sections. My Data allows you to change your account passwords. Whenever you submit a new file to PaintOmics, files are automatically stored in your personal space so you can reuse them in future analysis and download and visualize them online. As you run new jobs in PaintOmics, My Jobs section will show the status of your jobs. You can resume your jobs and continue working or download the results after each process. Please note that the available cloud space is 200 megabytes. Use it carefully and remember that you can always delete old files to free space. The Supporting Tools section includes a brief introduction to how region-based omics and regulatory omics work. The Resources section includes several PaintOmics tutorial videos, documentation, and a list of example data. If you want to cite PaintOmics, please go to the Publication section and view our newest publication. We would love to hear from you. Please use the Contact section if you have any questions or suggestions about the application, and we'll get back to you soon. Now, let's look at how to analyze gene or metabolite-based datasets. Table 1A shows an example of a quantification file for gene and metabolite-based omic datasets. The first column must contain the feature name or identifier. Generally, PaintOmics accepts Entrez gene IDs as feature identifiers, although other identifiers or name domains are supported for some species. The remaining columns contain the quantification values for each sample in the experiment, preferably on a logarithmic scale. Table 1B shows an example of a relevant features file, a unique column containing the identifiers or names for all significant features in the experiment. I will demonstrate how to analyze gene and metabolite-based omic datasets using Statigra data, which collects multi-omics data for a mouse B cell differentiation process triggered by the expression of the Icaros transcription factor. Here is a screenshot of the Statigra gene expression data. The first column contains the gene name followed by the tab-separated quantification values for 6 different time points on a logarithmic scale. Differentially expressed genes are provided as a list. The metabolite data has the same format. Now, I will use PaintOmics to analyze the Stategra data. Step 1. Upload data. The main page shows a data uploading form with two sections. In the first one, a combo box is provided to allow you to select the organism from an alphabetically ordered list, supporting partial filtering by writing in it. If the species is not present, you can use the link to request it, and we will install it as soon as possible. The right side of the first section allows you to select analysis databases. For some species, more than one database might be available. You can click Supported ID and Databases for more detailed information. The second section contains two parts. In the right one, you can enter the input data for each omic to include in the analysis. For doing so, the Browse button opens a menu with three options. Upload file from my PC. A conventional approach to select the file from your computer. Use a file from My Data, only available for registered users. Upload the data from the personal cloud space. You can use Upload New Files section to upload data to cloud space. Clear selection. Reset the selected file, if any. By default, gene expression and metabolomics, omics are provided, but on the left side, you can add more by clicking the icon. Similarly, the icon allows you to undo the action. An important setting is a type of enrichment to perform, based on genes or features. If no selection box is visible, like gene expression or metabolomics, it is because PaintOmics assume a default type. The selection might affect the contingency table used for Fisher's exact test in the pathway enrichment. For instance, in proteomics data, one protein identifier may match more than one gene identifier. In the feature enrichment type, the protein would be counted once, whereas, for the gene enrichment type, the amount would be the number of genes matched by the protein. Once everything is ready, press the button Run PaintOmics in the upper left corner. Step 2. Matching summaries and metabolite assignment. A summary panel shows the mapping summary results by each available database when multiple databases are available. PaintOmics processes your files and offers you different plots showing the results of matching the identifiers of your files to the KEGG, Reactome, MapMan ID. You can also view diverse information about your data by hovering over the data distribution plot with the mouse cursor. PaintOmics will calculate the clusters present in the data provided for each omic. It will use the method k-means, which uses an automatically calculated number of clusters are the ones you define here. Two additional sections would be available if metabolites-based omics were provided. The first one is the metabolite class activity threshold, where a binomial test is used to assess the hypothesis of the proportion of significant compounds in a given measured metabolite class being higher than a user-defined threshold. If you do not define an activity threshold, PaintOmics will use the average percentage of significant metabolites as the threshold. Another section is to review the assignment of metabolites identifiers and correct it. Each compound found in your data will show many checkboxes with the candidates. You can manually correct any errors for the assignment. When satisfied with the settings, click the Next Step button. Step 3. Visualizing the results. In the final step, PaintOmics displays the obtained results, offering many configuration options for tweaking their visualization. Pathways Summary. This panel shows you the job ID and the link you must use to access it. If you want to share your result with others, please go to the Sharing options and click Allow Link Sharing. By default, all jobs created by a user account are private, and the others are public. You can also view the number of found pathways and the significant ones based on each database if multiple databases are available. These counters will be automatically updated based on your filtering options and p-value selection, using the Fisher combined method in cases with more than one omics by default. The next section is Pathways Classification. Each pathway is associated with a primary category and subcategory, and you can access that information by expanding the tree elements. You can show or hide individual nodes by clicking on the checkboxes, or entire branches by clicking on the links that appear when hovering over the options. Then the Apply button. You can switch to different databases if available through the top button. The Pathway Interaction Network is built according to the process described in the previous PaintOmics Concepts video. Now, it is Metabolite Hub Analysis. If gene expression and metabolites-based omics are both available in the input dataset, a separate table for metabolite hub analyses will be shown. The Metabolite Hub Analysis provides a graphical assessment of the magnitude of differential expression for linked genes and metabolites in a heat map. You can easily filter the hub metabolites through the drop-down menu in a given step. Clicking on the Paint column icon will load the detailed expression information for both hub metabolite and its neighboring genes. Click Search icon will filter pathways only including the hub metabolite. The Metabolite Class Activity panel will be available if the user provides the metabolomics data. The panel shows the enrichment result of a metabolite class, and the Paint column icon will load the expression details of all the metabolites inside the metabolite class. In the Pathway Enrichment table, you can see every pathway with at least one match with your data. The table's header has some filtering options to search through the table quickly. Note that this live search is not permanent, will not be saved, and will not affect the number of found and significant pathways, unlike filtering by category as explained in the Classification Panel section. The visible columns can be individually adjusted by hovering the column block, then clicking on the arrow that will appear on the right side. Depending on the job, there can also be some additional select boxes to change the method for adjusted p-values or combined p-values. In the last case, selecting Stuffer will enable the Configure button, allowing to use of custom weights for the calculation. For reactome pathways, an additional column reactome class p-value is available, which enriches the reactome pathways based on its class. This aims to solve the issue that some reactome pathway maps contain many features, and some do not, leading to an enrichment issue for pathways without enough features. Clicking on the Paint icon will load the detailed pathway view. The main component of the detailed pathway view is the diagram showing all the nodes and the relationships between them. Matches are represented by boxes painted over those nodes, containing a heat map with the record values. One box can have more than one gene or feature associated with it, symbolized by a plus icon. If at least one of these features is significant, a star symbol will also be visible. Placing the cursor over these boxes will open up a tooltip window, expanding the info. If multiple features are associated with it, a previous, next link will appear in the lower part of that window, allowing it to iterate over them. Now, let's look at the region-based omics. Omics approaches for studying regulatory aspects of gene expression, such as chip sequencing or ATAC sequencing, typically return potentially functional regions, defined by genomic coordinates, which must then be related to proximal genes in order to gain any biological meaning. Therefore, integration of those region-based omics requires an extra step where regions are associated with genes based on their relative position with respect to specific areas of the gene. For example, ATAC-seq is a method to investigate the accessibility of chromatin, which is a method to determine regulatory mechanisms of gene expression. Therefore, it can help identify promoter regions and other regions, potential enhancers and silencers. To achieve this objective, PaintOmics 4 incorporates RG-match, a rule-based and highly configurable method for computing region-gene associations, annotating each associated with the area of the gene where the region overlaps. In order to compute the associations, Regions 2 genes needs the following information. A GTF annotation file contains the chromosome positions of all the features to be considered. This GTF file must be sorted and should include annotations at the exon level. That is, the third column of the GTF must contain the exon tag. A modified bed format file containing the regions of interest to be associated with features, followed by quantification values for each region. A list of all the regions is especially relevant for our experiment. By default, the region-to-gene associations will be computed as follows. The maximum distance of feature associations will be 10 kilobits upstream or downstream. The transcription start site area will start 200 nucleotides upstream of the transcription start site and will end at the transcription start site. The promoter area will have a length of 1300 nucleotides. Thus, it will start at 1500 nucleotides upstream of the transcription start site and end at 200 nucleotides upstream of the transcription start site. All possible associations and areas will be reported. This means that if a region overlaps several areas of a given gene, all of them will be returned. The next section is the regulatory omics. The upper table shows the example of regulator expression files. The first column must contain the identifier of the regulators. The remaining columns contain the quantification values for each sample in the experiment, preferably on a logarithmic scale. The upper left shows an example of a relevant features file for regulators. A unique column containing the identifiers or names for all significant features in the experiment. The lower table shows the example of the association file. The first column contains the identifier of the regulators, and the second column contains the feature name of the targeted features, gene name, metabolite name. See above the accepted format for the file. The relevant association file is a subset of the association file that could be directly uploaded by the user or automatically selected using correlation in PaintOmics 4. The relevant features uploaded through the relevant regulators file will show a red star inside the pathway visualization. In contrast, the features in the relevant association file will show as the yellow star inside the pathway, no matter whether they are directly uploaded by the user or automatically selected by PaintOmics 4. Here is the example shows the visualization of the regulatory omics inside the pathway. The feature with a yellow star symbol indicates a relevant association between the feature and its regulators, and the feature with a red star symbol indicates that its regulators are significant.